Thanks everybody for joining us virtually. It's really exciting to be able to have a way to still showcase and celebrate our undergraduate research projects that our undergrads have done for the applied ecology minor. So in case you're not really familiar with everything that our students have done, in order to kind of earn and complete the minor requirements, you have to take general ecology, so that's applied ecology or plant biology 360, and you have to take our capstone applied ecology class, AEC 400. You also have to complete 2 electives of the student's choice, which is very broad, right? As applied ecology can be interpreted very broadly. And in addition to those 4 course requirements, students also have to complete at least 3 credit hours, which is 135 contact hours on an independent research project that they have developed with the help of a mentor, right? And that mentor could be faculty in applied ecology, faculty in any other department at NC State, faculty or grad students or postdocs at any university outside of NC State. And we have even had students complete these independent research projects in partnership with an agency or a nonprofit or a startup before. So the world is your oyster, right? It really is limited only by the imagination and the individual pursuits and opportunities created by these students for themselves. So this minor, the research component, right? This research project is developed by the student and their mentor. And the idea is to get the full experience of the scientific process. So together you have developed a scientific question and you have targeted specific data that you need to collect using fieldwork or using meta-analyses in a digital data analysis format, right? So you've collected data, analyzed it, and then applied it back to see whether or not your hypotheses may have been supported and how this answers your scientific question. So all that to say, congratulations and very well done for completing this extensive project. I'm really excited to have 10 students with all very unique questions that y'all have addressed across departments and across systems. So getting us started today, we've got April Sharp, a junior in environmental science with a concentration in entomology, who worked with David Tarpey.
All right, good morning, everybody. Today I'm going to share with you my research on using pollen grain identification to document spillover pollinator visitation. And this project was an extension on Hannah Levinson's study on pollinator plots and their effects on soybean agricultural ecosystems. So bees are very important pollinators. 87% of flowering plants require pollination, and 75% of crop plants require pollination, the majority of which is done by bees. So the pollinator efficiency of different species of bees varies. This graph from a study from Shelley Rogers demonstrates that. She compared the pollinating efficiencies of some different species of bees and their pollination of blueberries. Now, it's important to note that blueberries do require pollination from insects. And there are some crops that are self-pollinating and they don't require insect pollination, an example of which is soybean. So even though soybean is self-pollinating, we still know that bees visit soybean flowers. And soybean is a non-model crop species in the context of pollination, since there's kind of a gap in knowledge of what role bees play when they visit soybean flowers. So that introduces a question of what role do bees play in the interaction? Do they contribute to pollination and cause an increase in crop yield at the end of the season? Or are their visitation merely having no effect on soybeans? So there's some questions that I'm hoping to address through the study. How does bee visitation affect the pollination of soybeans? How does the addition of planted habitats such as pollinator gardens affect bee behavior and agricultural ecosystems? And what are the long-term effects of large monocultures of self-pollinating crops on pollinator populations? So soybean is one of the highest yielding crops in the United States. 1.8 million acres are grown in North Carolina and 90 million acres grown across the United States in total. So for some background information on the pollinator plot project, here's an example of the experimental design of a site. There's 2 fields of soybean and a pollinator plot. One of the soybean fields is close in proximity to the pollinator plot while the other one is farther away. Here in this example, the close soybean field is 30 feet from the pollinator plot, while the far one is 0.3 miles away. And this same experimental design was more or less replicated at all of these research stations shown in this map. I, for this project, chose 3 of these stations to examine the relationship in pollinators based on the spatial regions of North Carolina, the mountains, the Piedmont, and the coastal regions. So some objectives of my study, my hypothesis is that higher pollinator visitation will occur in the soybean field closest to the pollinator plot. I predict that a larger pollen diversity will be found on bees collected from the near field, and pollen on the bees will reflect the pollen found in the pollinator plot. Some applications for the study assessing the benefits of patch habitats is both as refuges and sources for pollinators and agricultural ecosystems, as well as assessing what role bees play in soybean. So in our experimental design with the 2 soybean fields, we collected bees from both of those soybean fields, as well as a pollinator plot. The species that I sampled in this study were Apis mellifera honeybee, shown in the picture to the left, as well as Bombus impatiens, a common species of bumblebee in the middle, and Xylocopa virginica, a common species of carpenter bee, which is on the right. I chose these 3 species because of their abundance throughout North Carolina, and there were some of the major species collected in each of our 3 sites that I looked at. And they're pretty representative of some of the bee species found in North Carolina. We also collected flowers from the pollinator plots, and I analyzed them for pollen by removing their anthers and dyeing them on a microscope slide so I can identify their pollen characteristics and be able to reference back to them later. So then I collected pollen from the bees. I collected pollen from 2 different areas on the bee's body, either the corbicula or other cetaceous areas of the bee where I could visually see pollen, and I dyed it on a microscope slide. Then using my microscope, I selected 6 fields of view at 400 magnification. I selected 3 fields of view per area of the bee I sampled, and I counted the pollen grains and identified them. So here's some of my preliminary results from the first site that I completely analyzed. Unfortunately, I have not been able to get to all of my sites due to the current situation with the pandemic, but here's a figure of my results from our site in the mountains. So the proportion of soybean pollen on the bees in the far field was higher significantly than the proportion of soybean pollen found on bees in the near field. The proportion of pollen in near field was pretty homogeneous from the soybean pollen compared to other sources of pollen, and the soybean pollen found on bees in the far field was much more mainly soybean and less other sources. So I found that bees from the far field had a greater proportion of soybean pollen on them compared to other sources of pollen. I also found that more variability in pollen composition was found on bees collected from the near soybean fields. All bees that I studied in this, I surveyed, harbored both soy and non-soy pollen, and 13 non-soy pollen sources were identified in total. 6 of them were traced back to the pollinator plot, and I have listed below their sources here. So this suggests that bees at both fields had visited the pollinator plot at some time during their foraging journey. This also indicates a higher spillover pollinator visitation in the near soybean field. I have this diagram to demonstrate that. More pollinators are dispersing from the pollinator plot into the near field compared to the far field, which does support my hypothesis. Some limitations to my study are that plant families display similar pollen characteristics, which makes them hard to identify and can be a confounding variable in my analysis. Also, pollen distributions on my slides may not accurately reflect that on the bees. Also, there's some spatial and landscape variables that might introduce some more confounding variables to my study. For example, there are more sources of soybean other than the near and far fields in each sample site. Also, my sample size was relatively small. So some of my next steps whenever I'm able to get back to the lab are to complete the analysis for the rest of my sites, also increase my sample sizes. I also plan to analyze the pollen composition of bees captured within the pollinator plot itself in order to compare what their pollen analysis is to those in the soybean fields. And I will be doing this with the aid of a grant from the College of Natural Resources, which I'm very excited for. And finally, I would like to thank Hannah Levinson and Dr. David Tarpey for their mentorship and guidance in this project. Also, Dr. Elsa Youngsid for help and pollen identification. And thank you, Dr. Erin McKenney, for organizing all of this. Also like to thank the NC State Apiculture Program, as well as the Department of Applied Ecology and College of Natural Resources. And thank you for listening. If you have any questions, I would love to answer to the best of my ability.
Awesome. Thanks so much, April. We'll imagine that everyone is cheering at the same time, right? A roaring crowd. So I think at this point, if you could stop sharing your screen so that we can see you. And at this point, I would welcome anybody who has a question to either enter it into the chat, or you can unmute yourself and say, you know, hey, April, this is Erin, I have a question about.
Great job, April. This is Becky Irwin. I enjoyed your talk. I may have missed this because I'm here with two kids as well, but can you tell me what were some of the types of bees that you collected pollen off of? Again, you may have said this and I may have just missed it.
Yeah, so I collected pollen off of Apis mellifera, honeybee, Bombus impatiens, and Zydecopa virginica, which is a common species of bumblebee and carpenter bee, as I'm sure you know. And I picked these because they were some of the most abundant throughout North Carolina and the different sites we visited. And I just thought they were very representative of bees from different types of habitat and foraging. And I thought they would create a encompassing representation of other bees that would be visiting the pollinator plots.
Great, thank you.
Thanks.
The compliments are rolling in on the chat box. I don't see anyone else with a question, but Michelle has reminded us to also recognize you, April, as our inaugural Dragonfly Pondwork Scholar. So Michelle has dropped a link in if anybody wants to learn more about that other aspect of April's research life. Okay, well, thank you so much, April, for getting us started. Next up, we have Jack Kurtz. He's a senior in computer science, and his mentor was Selena Ruzzi.
Good morning, everyone. My name is Jack Kurtz, and today I will be presenting on my independent research project for this semester. I studied how the body size of native Raleigh bee species has changed over time under Selena Ruzzi as a part of Dr. Rebecca Irwin's lab. But first, a little bit of background about myself. You did not hear Dr. McKenney wrong. My major is actually in computer science. But a few years ago, I was fortunate enough to take part in a research project for the computer science department called Nature-Inspired Computation, where I studied and built algorithms based on real biological processes, such as genetic algorithms and ant colony optimizers. This project sparked a real passion for me in this niche field. And so I decided to pursue a minor in applied ecology to really exercise this. And since then, I've been fortunate enough to work on some more cool crossover projects, such as my computer science senior design project, which was the Pollinator Garden Planner, an online tool to help homeowners build and test their own virtual pollinator gardens. I found one of the best parts of this minor is that it's really allowed me to pursue my other passions outside of computer science, namely bees. So when it came time to find a research project, I didn't know what I wanted to do at first, but I really knew that I wanted to study bees. So to lay some groundwork for the motivation behind my project, I'll pose the following question. Why study bees at all? Hopefully, everyone can think of a few reasons, of course, but I've found that it really helps to quantify this. So bees are important because they provide critical pollination services to 87% of angiosperm plants across the planet. As such, bees and pollinators in general are integral to maintaining strong biodiversity in many different ecosystems. Due to their strong impact, wide breadth, and sensitivity to the environment, many researchers and ecologists tend to use them as indicators for the health of an ecosystem. Furthermore, we can use body size to measure the health of bees and bee colonies, as it's been shown that body size has a direct impact on the amount of pollen load an individual can carry, which has further ramifications on the amount of pollination that occurs. Using time as our independent variable allows this study to be extrapolated into other studies. Researchers could use the bee size to measure the impact of other external factors that change with time, such as urbanization. And that's exactly what my research mentor, Selena, is working on, and how my project fits into the scope of Dr. Irwin's lab. So there is actually sort of a high demand for this specific type of data due to the reasons that I listed before, but for whatever reason, this data is missing. No one's actually taken the time to collect it, especially over long periods of time. Past research on this topic has also produced confounding results. In 2016, one team found that their native bee species were increasing in size with habitat fragmentation, whereas another team found that the native bees were actually decreasing in size with landscape simplification. So, pretty confusing results. And it should also be noted that each team only collected samples for 2 years, which really isn't a wide enough range of time to be assessed properly, as the changes in body size can be attributed to a lot of different factors. So that's where I come in. The objectives for my research opportunity were to measure the size of native bees over an extended period of time, specifically looking at how individual species change over time, which is something that's never been done before. Luckily, NC State is home to one of the largest collections of pinned bees in all of North Carolina, at the Insect Museum on the 4th floor of Gardner Hall. These samples, for the bees at least, date back all the way to 1900, so 120 years of sampling is definitely enough. Based on some of my preliminary readings, I formed 2 main hypotheses. My primary hypothesis was that the species body size would increase over time due to the urbanization of Raleigh. Urbanization leads to habitat fragmentation, meaning that bees would have longer foraging times, and having a larger body size would be more advantageous in both making these longer journeys and bringing back enough pollen for it to be worth the trip. My alternate hypothesis was that species would decrease in size due to landscape simplification and the loss of floral resources. And so my data collecting began. In order to accurately estimate bee size, I measured their intertegular distance, which has been shown to correspond directly with dry body weight. For those of you who are unfamiliar with this metric, the IT distance is this measurement between where the wings meet the thorax, also known as the tegulae. I accomplished this by hand using a handheld micrometer under a simple compound microscope. And as I've now learned, I have some pretty shaky hands. So for better or for worse, by consequence, I also learned different methods for pinning, gluing, and repinning and regluing insect specimens. I also had to record the metadata for the captured specimen, such as information about where they were caught, what tools were used to catch them, and who was responsible for catching them. Since we were primarily interested in the body size of females, it was important that I learn the difference between males and females so that I could identify all of the unlabeled samples. As it turns out, there are 3 main ways to distinguish between the males and the females. First and foremost, female bees tend to be bigger than their male counterparts, which makes sense because they are doing all the work after all. Secondly, and this is true for most species, the males tend to have some sort of yellow facial markings, whether it be hairs or just a yellow coloration on their skin that kind of looks like a yellow mustache, as you can see here in the images. Third, and probably the best indicator, is that males tend to have an extra antenna segment. As it turns out, when you spend hours looking at bees every day, you get pretty good at telling the females from the males. Before COVID-19 hit, I was able to measure 5 different species of bees: Andrena macra, Andrena miserabilis, Ceratina calcerata, Hoplites pilocifrons, and Osmia sandhousei. These species were mainly chosen due to their availability in the Bug Museum over a long period of time. But unfortunately, the university shut down and I no longer had access to the museum. So the latter half of my research revolved around the analysis of my collected data. I learned how to build a reusable pre-processing pipeline in R so that once I was done collecting data for a species, I could see results almost instantly. I learned about some different visualization techniques with R and the ggplot library, such as jitter and box plots. And finally, I learned how to perform statistical tests in R, such as looking for the correlation between 2 variables, such as the bee body size and the change in time. Unfortunately, for all the data that I collected, there really was no noticeable change in size over time for any of my species. The highest correlation coefficient I could find was negative 0.28, which is nowhere near significant. You don't even really need any heavy statistical tests to see this, as the line of best fit for almost all of my graphs is basically a fine. So as you can imagine, my results were kind of a major roadblock for me, as they were almost the exact opposite of what I expected. Both of my hypotheses were not supported by the data. And so my first interpretation of my results had me, a little bummed out. I kind of felt like I hadn't accomplished any work at all. So one of the things that I had to learn was that negative data is never actually a bad thing, and it's certainly much better than no data at all. Furthermore, the interpretation of no change in body size over time could mean that urbanization is not having an effect on bees at all, which from an ecological perspective could be a positive takeaway. At the very least, the data that I've collected can be used by my team and other teams in the future. So all that work was never really wasted. And of course, I just want to say a huge thank you to Celine Rusi, my research advisor, Dr. Irwin, my lab head, and the rest of the Irwin Youngstead lab group for all their help and support along the way, especially through a crazy time like this. And also a huge thank you to Dr. McKenney, not only for planning all of this and the research, but for all of her help along the way in the applied ecology minor. My goal at the beginning of this semester was to really just gain my first experience in a lab with a research team. And I'm definitely leaving this semester with a new set of heavily applicable skills for my future. Thank you for your time. I'll now take any questions.
I have a question. This is Becky. Great job, Jack. I enjoyed your talk. Because your data collection got cut short with the virus, were there any species that you were still planning to measure, and what were they?
Yeah, there was one more species that we were planning on measuring. It was Caliopsis and Andreniformis. That was the last one on the list.
And I see, Rob, you've got a question. Do you want to ask it yourself?
Sure. My connection is never great, so I'll try. But what other features of these bees do you think, imagine you could measure anything going back through time, might have changed? So thinking about diet or their microbiomes or their behavioral interactions or genes. Mm-hmm. What else might you want to look for going back? I don't know the answer. I'm just curious.
Yeah, I think one of the big ones for me would have diet, because like one of the biggest changes that I expect over time is the introduction of different urban gardens, and I think it'd be really cool to see how those the introduction of new plants from humans may have affected their body size or their change in diet.
Thank you.
Huge thank you, Jack. That was really great work. And up next, we have Haley Shoptaw. She's a senior in environmental science, and her mentor was Katie Martin. So Haley, feel free to take it away.
Alrighty, hey everybody. As Aaron said, my name is Haley Shoptaw, and I'm a senior in the environmental science department. And I'm going to be the first non-bee person of the day. And I'm going to be talking about the remedial effects of an urban forest on macroinvertebrates within an urban watershed. So to start off, a little bit of background information about macroinvertebrates in general. Macroinvertebrates are typically the larval stages of various insects, and there's a ton of diversity in freshwater ecosystems of benthic macroinvertebrates, which were what I was looking at. So I'm going to be clicking through some pictures of different groups of macroinvertebrates based on their pollution tolerance. So macroinvertebrates are typically the base of the food chain, and they support the rest of the ecosystem, either through being a food source for other organisms or through the ways that they themselves eat. So they're based, or they're grouped into groups of either shredders or collectors, grazers, and predators. That's their main 4 ways of eating. And the ones that I mostly found were shredders, grazers, and a couple predators, which were mainly dragonflies. They're typically about the size of a penny, and they're frequently used as a bioindicator of water health, as evidenced by these pollution tolerance categories. So there's also a few different criteria that have been well established for using macroinvertebrates as bioindicators of water health. One of the most common ones is the EPT test, which looks at 3 different orders of macroinvertebrates, being the Ephemeropta order, the Plecoptera order and the Trichoptera order. And that would be the three included in the EPT test. And I have pictures of 3 examples of members of those orders being mayflies, stoneflies, and caddisflies. And luckily, I actually got to see all 3 of those in the stream that I looked at. And if you look at the bottom of this slide in the Lynat 1988 table, you can see the levels of, or the numbers that you would hope to see in each of those taxas to classify your water through excellent and poor. So a little bit about the area that I studied in general. So I was in the Carl Alwyn Schenck Memorial Forest, which is under that big red circle, which is in the middle of the Richland Creek water basin. So the rest of the surrounding area is mostly urban. It's in the middle of Raleigh. It's right by NC State University. And it's about 20% impervious land coverage, according to the USGS. So zooming in on the Carl Alwyn Schenck Memorial Forest in general, it's right beside I-440 and Wade Avenue, which was really important to my study. And there was a clear cut scheduled in January, which is shown with that yellow cross section. So my initial plan was to sample within streams 1, 2, and 3 labeled on the map before and after the clear cut to see how that clear cut affected the macroinvertebrate communities. Unfortunately, that didn't work out as streams 2 and 3 were dried up. And so instead I focused on stream 1. And I found 6 sites within stream 1, which runs right under Wade Avenue. So I started looking at how the forest was helping the macroinvertebrate communities recover from whatever pollution Wade Avenue or I-440 were dumping into the stream. And each of those sites, sorry, let me go back one second. Each of those sites are about 150 meters apart as the crow flies. So it was a pretty even spatial analysis. So my hypotheses, my null hypothesis obviously would be that nothing changes as far as macroinvertebrate pollution dynamics, regardless of how far you go into the forest. And my alternate would be what I expect to see, which was that the macroinvertebrate populations would be larger and that you would see more pollution sensitive species as you move further into the forest. So how I went about doing this was I took kicknut samples at each of those 6 sites that you saw before. I took 2 kicknut samples at each site. In the kicknut, you can see in the bottom right was a cloth bag with a mesh lining in the middle. I stuck the kicknut perpendicular to the flow of water and agitated the surrounding area for about 60 seconds, and then dumped whatever I caught in the net into a tray for sorting. You can see pictures of some of the bigger guys that I caught. There was a crawfish in the bottom and just a regular fish in the top right. I didn't collect those, but everything else that was too small to identify in the field was put into a 70% ethanol solution so that I could transport them to the Ardone lab. And once I was in the lab, I placed the macroinvertebrates under a dissecting scope, which you can see what I saw through the dissecting scope in the bottom 2 pictures, and then compared them to river watch guides, which there's actually a picture of one of the watch guides that I use the most right in the center here, which shows kind of the different characteristics of caddisflies, which is pictured in the bottom right. So the hooked legs, the 2 tufts at the back. And then I use those same river watch guides to record their relative pollution sensitivities. And so this is a picture of what that data looked like. It's a bit messy because there was a lot that went on in my study, but I just recorded the number of individual macroinvertebrates I collected and color coded them based on what type of bug they were. There are a lot of blacked out sections on this graph or on this chart. So the first sample was the day that I went out and was initially hoping to sample in streams 1, 2, and 3, pictured on the bottom right. But as you remember, streams 2 and 3 were dried up, so that didn't work out. So going out for my second day of field work, I changed gears and went to 4 sites within stream 1. I was still hoping to look at the clear cut effects, but after talking with my mentor, we realized that I wasn't going to be able to see that as well as I wanted to. And so I went back and added sites 0 and -1, which are right beside the highway, so that I could start to see a larger spatial scale of the highway's effects. And so this is what that looked like. As you can see, I have 2 pretty major outliers indicated by these big red arrows. Most of the sites were maxing out around 10 to 15 macroinvertebrate individuals per site. And then site 3, which is the big orange bar, is seeing up to 116. So I had to adjust my graph for outliers so you can actually see what I saw, which is here. So as you can see, there wasn't a ton of variation between each of my individual sites, except for site 3 being massive. And then in January, I had a huge drop off in the number of macroinvertebrates that I was collecting per sample day, which you can see in my February and March samples. So looking at just all the average, or all the numbers at each site over the entire breadth of my study, you can see that I saw a pretty nice little curve up to site 3, and then site 4 drops back down to about the same level as site -1. And I'll talk a little bit about why that might be in a second. But each of these arrows indicates statistically significant differences, which were determined by a one-way ANOVA test followed by a Tukey's test. So site 3 was significantly statistically different from every other site on the map except for site 2. And then site -1 was different from sites 1 and 2, but not site 0. And site 0 was different from everything except for, everything except for site 1. So we did see some good differences between sites, but we didn't see them among sites, and that's something I'm going to talk about right now. So within each site, there was no statistically significant difference between the number of pollution-sensitive species or intolerant species or tolerant species. So I had to throw out that part of my alternate hypothesis. And then I looked at other studies that had done similar work and saw what sort of numbers they were looking at total macroinvertebrate individuals, which is shown in this red box, is the total number of macroinvertebrates collected actually in Raleigh, in an urban Piedmont area of North Carolina, in a Lannat study in 1994. As you can see, the lowest number they ever saw was 199. And the largest number I ever saw was 116. So unfortunately, due to how low my numbers were compared to other studies, I can't reject my null for that part either. So right now, my null hypothesis stands. So a little bit about why that might be. As you might remember from my graph, I had these two huge dips right before my February sample and right before my March samples. And those were probably caused by a large storm that we had and a snowstorm that we had, which you can see on the calendar, how that falls between all of my samples. And then in the graph on the right, how that falls in between my samples, where you can see the drastic decreases. And so that probably washed out a lot of the macroinvertebrates that I was looking at, or just damaged the ecosystem in general. And I'm really lucky, a faculty member in the Applied Ecology department, shout out to Michelle Jewell, has a 10-second time lapse of the storm that happened on February 6th. So we're gonna look at that real quick just to see how drastic it was. So as you can see, it was a lot of rain. We were actually under a tornado watch for most of the day. So it was a pretty bad storm. And after that, this is what my trails looked like. So if you look on the bottom right, the black dotted line are the trails that I was walking, and the big red circle is where site 4 is. So that could possibly explain why site 4 was performing so much worse than every other site, is because it's right at the base of the trails. And so any runoff water would have ended up right in site 4 and could have really hurt macroinvertebrate communities. And then part of how those storms could have really hurt the macros are, this picture up on the left is actually the trail marker. You can kind of see the little hut with the green roof. And that's where the trail is supposed to be. So there was a lot of flooding and a lot of sediment washout. And so due to COVID, I was never able to go out again and see if my macros were going to recover from that storm. So all I have is a huge drop off after the storm. And that's all I was really able to find. I want to give a huge shout out to Dr. Katie Martin for taking me on after my class performances last year. She's had a lot going on in her life, and I've had a lot going on in mine, so I'm really grateful for her patience in working with me. Also very grateful to Dr. Ardone and his lab manager, Pete, for letting me invade their space and spill ethanol all over the place. And then a huge thank you to Logan and Will, who are pictured in the right, for helping me out in the field. They were real lifesavers. And that's all I've got. So if anybody has any questions, let me know.
Great job, Haley. Let's open it up for questions.
Yeah, so Dr. Gunstead just sent a question in the chat asking what I would do if I were to continue the project. I would try to collect more data on sites 0 and -1 since I added them so late in the game. There wasn't a ton of representation. And I would definitely do, if we're going to do it again, I would add more water analysis just to see what might be influencing macroinvertebrate community trends that I was seeing. And moving forward, I would probably add that as well, just because I'm guessing that sediment disruption is what caused that huge drop in the number of macroinvertebrates I was collecting, but I'm not sure. So I would love to measure things like turbidity and just like overall sediment load. What is the most important thing I learned from my work? For my future career, I've learned that I really don't like killing my research animals. And I know that that's kind of morbid, but I did have to collect all of my macroinvertebrates in a 70% ethanol solution. And as non-charismatic as they might seem to like the regular eye, after you work with caddisflies for months, you start to learn their little mannerisms and think that they're really cute. So that broke my heart. And so that's something that I'm going to carry with me for my future career. But overall, just from the experience in general, definitely the statistical analysis that I did were really helpful to learn. I'm going to be using them. I'm starting my master's next year, so that'll be very helpful to know. And just the whole procedure of literature reviews and creating my own methodology will be really helpful with my master's and potentially my PhD later on.
Awesome job, Haley. Thanks so much. I'm going to transition us over to Aiden Smith, our next presenter. She's a senior in marine science with a concentration in biological oceanography, and her mentor is Astrid Schnetzer.
All right, so my name is Aiden Smith. As Dr. McKenney said, I'm a senior in biological oceanography, and my presentation is going to be about examining cyanotoxin food web contamination in the Chowan River, North Carolina. So I'm not sure how many of you guys are aware, but last summer there were a number of headlines about an unusual degree of algal blooms occurring in the North Carolina coast. So a lot of dogs got sick, a lot of people were cautioned not to swim in the water, things like that. So if you're familiar with it, this was definitely a hot topic last summer. Harmful algal blooms are an increasingly common phenomenon due to a lot of conditions, temperature, increased nutrients in water, things like that. And severe bloom events in the Chowan River and western Albemarle Sound have been more recently associated with the presence of cyanotoxins. So if you look at this slide here, cyanobacteria is a naturally occurring blue-green algae, and sometimes in the case of harmful algal blooms, it will produce toxins like microcystin and cylindrospermopsin. So those are the two toxins that we were specifically looking at. And this is actually the first study of its kind for North Carolina estuary. So we are really excited about providing some really important information about these harmful algal blooms and the cycles. So overall, we wanted to determine the presence or absence of cyanotoxin movement through the food web. So at the bottom of this diagram, you can see the cyanotoxins, and then they would be produced and go into the clams that were filtering the water, or potentially absorbed by the fish, and then eventually they might even make their way into people who are eating the fish or the clams. So we wanted to look at that and establish a time series for algal blooms in the Chowan River and the Albemarle, as well as analyze trends in toxin accumulation, so which organisms we were seeing the most toxin in and where. So based on this map, we sampled at various locations along the Chowan River and the Albemarle Sound. It's an area that a lot of people use for fishing and collecting clams, and we have seen an unusually high degree of algal blooms in this area recently. So the first thing we did was collect finfish by trawling through May and into October. Initially, we started going on a weekly basis, and then it decreased to bi-weekly and then monthly. The top picture on the left is of a white perch. That's one of the species of fish we were looking at. And then the bottom left is a clam that we were looking at as well. Clams were collected from designated clam beds, so we weren't trawling for those. The fish were collected through trawling, and then the clams were just collected from the clam beds. And then both clams and fish were weighed. We measured the fish from the nose to the fork of the tail, and then the clams we measured from side to side of the outer shell. And then the tissue was cut into roughly 2-gram samples, and then the clam tissue was also cut into roughly 2-gram samples. After the samples were cut into our designated weight, we put 4 grams of methanol solution into them, and then we homogenized them, which is this picture on the left, to kind of create a pulp and break apart some of the cells, start that process. We then sonicated the samples, which is a piece of equipment that sends pulses of sound into the samples and further starts that cellular breakdown process. And then we went ahead and centrifuged them. After the centrifuge, the resulting supernatant was filtered into little cryovials. The cryovials were frozen until we were ready for analysis. And then we conducted an ELISA chemical test, which is an enzyme-linked immunosorbent assay to determine cyanotoxin concentration. So these are the results that we found. You can see a pretty clear cycle of toxin concentrations, mostly microcystin, which is in blue. It kind of starts pretty low in May and then peaks in June and July. You can see a decrease in August, and then into September and October, it's really decreasing, and then cylindrospermopsin starts to increase, and that's the orange that you can see on the tail end of this time series. So that was really interesting because we were kind of expecting them to be present at the same time, but based on this specific data, you can kind of see a cycle of when it starts to enter the tissues that we were looking at, and then when it starts to decrease, and then when cylindrospermopsin starts to kind of take the place of the microcystin. And then this was the finfish that we were looking at. The previous graph was for the clams. The finfish overall exhibited significantly less toxin accumulation than the clams did. Maybe because the clams are filter feeders, we're not 100% sure. We're definitely gonna need more data to establish that. And then if you look at the gut content versus the tissue content, the toxin concentration in the gut content was significantly higher. And then again, you barely see any cylindrospermopsin, which is really interesting. And that's definitely something that we'll wanna look at in the future. We separated the samples into tissue versus gut samples to see if it was coming from what they were eating or potentially through gills and the bloodstream. Again, we definitely need some more sampling to look at what the highest concentrations are and why. So again, the clams showed significantly higher concentrations of cyanotoxins than the finfish. And then microcystin concentrations were overall significantly higher than cylindrospermopsin for both clams and the finfish. And while microcystin was relatively present in both clams and fish, cylindrospermopsin was not observed until later in the time series, closer to September. So establishing kind of a cycle would definitely be very helpful because this was kind of our first season taking this kind of data. So trying to figure out maybe sampling later into the months and after September and October to figure out what that cylindrospermopsin cycle looks like would be really helpful as well. And some of the pitfalls with this were difficulties with lack of data. So if you remember going back to the finfish diagram, there was basically no concentration of cylindrospermopsin in the fish. So it was kind of difficult to establish any cycles with that, which, to be honest, is great for the overall health of the ecosystem we were looking at, but makes it a little difficult for us to, manipulate data and figure out kind of what's going on in that area. And then the ELISAs that we use for chemical analysis, which are pictured here on the right, are really sensitive. There's a lot of micropipetting that has to go on, and if there's any cross-contamination at all, it can completely ruin the whole plate. So that was definitely really difficult. Learning how to do that was helpful and definitely good experience, but a long process, and you had to be really, really careful. And even with triplicate data, it was really difficult to find repetition of values as well. So in the future, we want to do some more studies over a period of multiple bloom seasons, because this will help us understand more about the cyanotoxin impacts through the food web and how it's moving, where it's accumulating, and then other patterns of production and sequestration that might occur. And then the research was actually funded through the Chowan Community Research Grant. And this is an ongoing program, so hopefully we'll be able to continue this into late summer of 2020 and possibly into the fall. Depending on funding, we'll see how that goes given the current situation. But this is definitely a common and a common issue along the North Carolina coast. And then acknowledgements, Dr. Astrid Snetzer is my PI and my advisor. She was wonderful, helped a great deal with this. Marco Valera is the plankton ecology lab manager and taught me how to use all of these different materials and all of the sampling techniques and stuff, so he was really helpful. And then Emily Griffith is another undergraduate researcher who helped with a lot of the data analysis and dissections and things like that. So those are the three main people I wanted to thank. But overall, this has been a really great project, and I look forward to seeing where it's going to go. Any questions?
Joanne, did you have a question?
Yes, I did, but I'm not sure who, is everybody still there?
Yeah, I'm here.
Hi, great job, Aiden. I enjoyed your talk. Is there any information about whether cyanotoxins in these areas are affecting fish health or invertebrate health in any way, or is this all pioneer?
So this is pretty pioneer. We've done a lot of research previously, both in this lab and the North Carolina Department of Environmental Quality, with suspended toxins that are actually in the water column, but we haven't really looked at how it's making its way into the organisms. So that's kind of why we decided to do this project, because we wanted to look at how it was impacting those species.
Thank you.
Up next, we have Alex Hornaday, a senior in environmental science, who is mentored by Marcelo Ardon.
Hello, everybody. Right, so my project was investigating the effects of road salts on greenhouse gas emissions coming from urban streams. So when I say road salts, I'm referring to something that we're all very familiar with, even though we do live down here. It's what they spread on the road before and after snow events to reduce the effects of ice on the roads. So as you can see by the graph on the left, yearly US salt sales have increased a lot in the last 70 years. All the fluctuations towards the right is dependent upon how severe the winters are. And so these are obviously very prolific in the US. And so basically what they are is they can be in the form of a brine, which is usually applied to the road before a snow event to lower the freezing temperature of water. And they consist of an alkaline earth metal ion, like sodium, potassium, magnesium, and it's bound to chloride. And pay special attention to the picture on the right. What we have here is some salt as a part of runoff leaving into storm drains. So that is a big problem because road salts increase freshwater salinity. So in this graph, we see a very succinct picture of what happens. As snow melts, the runoff kind of seeps into freshwater bodies and carries the road salts with them. This leads to really significant salinity or salinization and alkalization of freshwater bodies. Sujay Kaushal is a really prominent researcher in this field, and he spearheaded a lot of research involving the salinization of freshwater and has really brought it to prominence. He has articles in the Atlantic covering his work. And so this is becoming more and more of an issue because we already have a good stock of the effects of road salts on water quality, flora and fauna, aquatic flora and fauna, and a little bit on freshwater biogeochemistry, which is something that we will go over in a bit. So streams are a significant source of greenhouse gas emissions. This is a graph acquired from my mentor, Dr. Rodon's research paper from 2011. And you can see that the numerical values represent petagrams per year of carbon from a terrestrial source going through freshwater bodies and then on the way to the ocean. Notice that a very significant amount of that is released into the atmosphere. And as most of us know, as it's released into the atmosphere, these greenhouse gases perpetuate climate change. And so this, combined with how prolific the salinization of freshwater bodies are becoming, led us to this experiment to test whether road salts, which is a very common human source of salinity, will have a noticeable effect on greenhouse gas emissions. So here are our objectives, pretty cut and clear. We wanted to correlate the use of deicers to increases in greenhouse gas emissions from freshwater streams, urban streams in particular. And so our original methods were to go out about weekly to the Rocky Branch Creek on campus. I'm sure a lot of you are familiar with it, and gather water samples to take back to the lab. I spent a good amount of time making these microcosm setups, which is a design given to me by Melinda Martinez. And so they're very basic, but really capture a very well organized portion of the microbial community that creates these greenhouse gases in streams. It was a mason jar. We filled it up with the stream bed sediment and then filled it up with stream water in 3 reps: a control rep, a rep that had a specific conductance of 300 microsiemens, and another rep that had one of 600 microsiemens. We drilled a hole into the lid and inserted rubber septums into there, because through these we would attach a gas met probe, which would measure emissions, specifically in methane, carbon dioxide, and nitrous oxides, which are all greenhouse gases. We were going to try to record these in 1 day, 3 day, and 7 day intervals and get a really good set of data to build upon. So, a pandemic hit, which we weren't really planning for us, and it actually hit right before I was planning on going out and getting the data, which is a shame. So the new methodology became the literature review. I went on Google Scholar and a few databases, and I kept my search limited to certain keywords: salinization, freshwater, stream, urban stream, greenhouse gas emissions, what have you. And so that became our method moving forward. So here are some quantitative results from my literature review searches. This comes from the Environment Complete database, which is a really large database hosted by EBSCO. I kind of chose it to represent the total pool of knowledge, or at least try to, because I think we all know Google Scholar can yield results that really don't have anything to do with what we search, so I didn't want to include those numbers in here. So yeah, as you can see, as my search became more specific to our question, the results became much more limited. There's a really significant body of research on, again, the effects of salinization of freshwater, but as you get more specific and incorporate, okay, which one of those effects perpetuate emissions? How do road salts play a part in it? We get much less results. A really good study that I found was done by a lab from Western Michigan University. It was a microbial ecology lab. So basically, they found that road salts can inhibit lake turnover, which leads to more methane emissions. So turnover as a concept, a lot of lakes are dimictic, which means they have turnover events in the spring and fall. And this basically happens when warm water, or cold water rather, which is denser than warm water, kind of sinks into the bottom of the lake, causes the warm water to rise, therefore mixing the lake up. This is really important for the distribution of nutrients and for something called the redox stratification gradient. This gradient is the settling of certain oxidized and reduced materials that almost create a kind of an impenetrable layer on usually towards the depths of lakes. And so gas can't really get through that, and things like air, methane, carbon dioxide cannot penetrate that layer. So introduce road salts into the equation. Basically, the road salts simulate density where it doesn't happen and causes water just to stay at the very bottom. None of that turnover happens. So what happens with that is the redox gradient gets higher and higher, and so it creates a persistent anoxic zone in the lake. In this zone, the bacteria produces methane, which concentrates at the top, can't penetrate that gradient, and eventually it emits into the atmosphere. On average, they found that these lakes under salinized conditions emitted about 3 times more than the control lake that they were visiting. So very, very significant effect on that. So what does this tell us? That study was very succinct, very thorough, and super helpful, but it wasn't specific to our system. You can't really apply concepts in lake hydrology to urban streams, especially turnover, not a phenomenon that we observe in streams. And so the search continues. And in the end, this experiment would have been best done with concrete data. But again, we had to make do with what we had. I think there's a lot to be said about larger scale research with wetlands. There is a good amount of research from several researchers about the salinization of coastal wetlands as a result of saltwater incursion. And so they simulated a lot of, Dr. Marcelo Ardon simulated changes in greenhouse gas emissions as soils became inundated with salt water or became more salinized. In general, depending on the condition, it was very dependent on the condition of the soil. Greenhouse gases changed according to predictions, not according to predictions. And the nature with tidal salt water is kind of different from road salts. So it's kind of hard to expound on that and apply some of those results to what we're asking here. So in the end, there is a need for more specific research to this question, and it is a novel question. So that's pretty exciting in the grand scheme of things. So we're happy to have been able to do that. That's it. Acknowledgements, of course, my mentor, Dr. Ardon, and this would be possible without him, Dr. McKenney for organizing this. A grad student with Dr. Ardon's group, Melinda Martinez, who gave me the design for the jars and walked me through it. And then Pete Lazar, the lab manager, because without him, I wouldn't be able to get into the lab in the first place, and that would have been problematic. So yeah, that's that.
Thanks, Alex. If you could stop sharing your screen so that anybody who has questions can, feel like they're interacting with you face to face. And we now invite questions for Alex. Feel free to type them into the chat feature or to unmute yourself and speak directly.
I'll just unmute myself. This is Rob. Really enjoyed the talk. Are there species that you would predict would be favored in the streams that end up with more salt? Or are we favoring different sets of species in addition to whatever is happening to?
To be honest, I didn't really account for fauna. It was really a hard study on greenhouse gas emissions. Are you talking about in terms of like bacterial species or
yeah,
just
anything? Are we tipping toward one sort of stream ecosystem versus another?
Sure. I mean, again, it's research specific to stream ecosystems, and salinity is kind of limited. But I would imagine that macroinvertebrates that are able to tolerate higher conditions of salinity would have a bigger niche to fill as streams become more salinized. So I'd imagine those would thrive in salinized conditions. Certain plants, perhaps, that have a high tolerance for salt as well, if that makes sense.
Yeah, cool. Thank you.
Yeah, of course. Good question. Thank you.
Alex, this is Marcelo. Nice job there.
Thank you.
So I guess one thing I wanted to follow up from Rod's question and your answer is, one way to think about it is that we could be starting to, in urban streams, we could be starting to create streams that look more like coastal streams. So we're kind of moving the biota and the streams in the cities to be more like coastal streams.
Mm-hmm. That's true. I guess you kind of be able to compare the ecosystems of certain tidal streams and coastal streams and kind of extrapolate those results and maybe test to see if they have a place in our urban streams under salinization conditions.
Yeah, and I guess another question I had for you, I mean, unfortunately, we didn't get to do the lab work we really wanted to do, but what did you gain from this? Do you feel you have a better handle of what research is like now, even though we didn't get to complete the research as we wanted to?
Yeah, I'm really good at drilling holes in jars at the very least, but no, definitely. I think it was such a holistic process when we started. I mean, we started with just the question, really. The sampling vessels were homemade. I learned how to use the gas meta part of this. We were going out into the field regularly. And I think it was just a really good introduction to a holistic research experience.
Great job, Alex. Thanks so much. So up next, we've got Nora Skinner. She's a senior in marine science, and she was mentored by Joanne Burkholder and Ellie Allen.
So my project was on the winter-spring shift in phytoplankton at Lake Johnson in Raleigh, North Carolina. So for those of you that don't know, phytoplankton are microscopic aquatic algae. Phytoplankton are only microns large in size, and they inhabit marine and freshwater bodies. And they're an important base of aquatic food webs in both. Phytoplankton are photosynthetic and are responsible for producing around 50 to 85% of the world's oxygen. Although the majority of phytoplankton are phototrophic, there are some species of phytoplankton that are mixotrophs, meaning they combine both phototrophy and heterotrophy. And then finally, zooplankton are heterotrophic plankton that I'll mention later on in the presentation. So right now you might be thinking, okay, phytoplankton, why do they matter? In addition to being an important base of aquatic ecosystems, phytoplankton are sensitive to water quality changes, and they can serve as bioindicators of nutrient and contaminant changes. And sometimes their reaction to these nutrient and contaminant changes is to form noxious blooms, which can leave water unsafe to drink or to use for recreation. It's important that we know which species of phytoplankton have the capacity to bloom so that we can monitor water bodies throughout the year and prepare for bloom events. So lakes in North Carolina, which are often just artificial reservoirs, are classified by productivity, which is referred to as the Trophic State Index. North Carolina lakes vary widely in trophic classifications, and most lakes are considered mesotrophic to eutrophic, which is moderately to highly productive. And previous research on eutrophication in reservoirs in North Carolina indicates that high nitrogen and phosphorus supplies often promote cyanobacteria blooms. So this is my study site, Lake Johnson. It's a small reservoir in Raleigh that is heavily used for boating and fishing. North Carolina reservoirs are highly individualistic in phytoplankton assemblages, which can be attributed to differences in things like the size and the turbidity of the reservoir, or even zooplankton grazing. And Lake Johnson's phytoplankton assemblage had not been studied previously to my project. So just a little more background. Previous research has shown that the general trend in phytoplankton succession is that we tend to see diatoms in the winter, chlorophytes in the spring, and cyanobacteria in the summer. So my goal for this project was to assess how the phytoplankton assemblage changes seasonally in Lake Johnson and whether that change is influenced by nutrient levels. And then my hypotheses were that as winter progresses to spring, there will be a shift from an assemblage dominated by diatoms to dominance by green algae, and then also that phytoplankton abundance would be positively correlated with nitrogen and phosphorus supplies. So what I'm calling phase 1 of my project was the actual phytoplankton, zooplankton, and water quality field collection. Once every month, I would travel to Lake Johnson, and I stood on the end of the boat dock to gather my samples. I would use a large water pitcher and scoop surface water off the side of the dock, and then I would pour the water into numerous vials. And each vial or bottle was for sampling a different water quality or nutrient parameter. And in order to test for some of them, like total phosphorus, they would need to be preserved. So for total phosphorus, I would drop 6 drops of 50% sulfuric acid in the vial and then leave the sample on ice. And I also collected a vial for a sample of preserved phytoplankton, which would be preserved with 6 to 8 drops of acidic Algal solution. And along with preserved samples of phytoplankton, I also took unpreserved samples. And for that, I would use a phytoplankton tow to gather a concentrated amount of phytoplankton, which is the tow that you can see on the slide. And I would stand on the dock and throw the tow out about 8 times each time I sampled to ensure that I got the same amount of phytoplankton for every month. I felt it was also important to mention the weather during my sampling period, as I'll bring it back up later. So record high temperatures were reported for each month during my sampling of January, February, and March. And most recently, it was reported that March 2020 was the 5th warmest March on record in Raleigh. And also from February 5th through the 7th, Raleigh experienced a heavy rainfall event where we received around 4 to 5 inches of rain. So moving into phase 2 of the project, phase 2 happened back at the Center for Applied Aquatic Ecology. And as soon as I would return from a sampling day, I would hand my samples over to the wonderful ladies in the water quality lab, and they ran test and quantified nutrient data for me. So then for me, it was time to get the samples of preserved phytoplankton ready to be observed under the microscope. I would have to settle a known volume of the sample in an Uter mold chamber, which is the tube that you can see on the slide. And this part always proved hard for me, and I actually ended up spilling quite a few number of the samples. But after the slide would settle for about 24 hours, then I would come back and I would count the chamber on an inverted microscope at 20x, and I kept a tally of each of the species that I saw. And I also took pictures of any cool phytoplankton. So phase 3 was the data analysis phase. In Excel, I would enter my phytoplankton data into a template that my mentor Ellie gave me, and then I would calculate the concentration of species and the percent presence of phytoplankton groups. And then in Excel, I also ran a linear regression analysis to see the relationship between log chlorophyll A and total nitrogen and total phosphorus. So our results for the phytoplankton assemblage in January showed that 8 phytoplankton groups were present, and the greens, or the chlorophytes, were in the highest relative abundance at 57%, followed by the diatoms in the orange, and then cyanobacteria. So these are just some pictures of some greens that we saw, some chlorophytes. We saw Enchistrodesmus, Sterastrum, and Spirogyra, and then this was just a cute picture of a little diatom. So in February, there was a similar relative abundance with greens remaining the highest, but then cyanobacteria were in a higher abundance than the diatoms. And this is a cool video, if I can get it to play, of a Malamonis actually being attacked by another algae species, which just goes to show that sometimes phytoplankton can be mixotrophs. All right, and then finally, things begin to shift in March with the onset of spring. The greens are around the same relative abundance as the cyanobacteria, and then of course the next highest is the diatoms. And here's just some more pictures. This was a cute chlorophyte Dictyospherium that we saw. We saw some cyanobacteria. And then I threw in this picture of this Trichlomonas, which is a euglenoid, just to represent some of the other groups that we saw. And just as an aside, we also looked at the zooplankton present in our samples, but we only had time to look at January and March. However, in just a month, there was a substantial change from dominance by rotifers to daphnids. And we know that zooplankton grazing can strongly impact phytoplankton biomass. So we just thought it would be interesting to look at the groups that were present in our samples. And here's some pictures. We saw lots of rotifers, copepods, and daphnids. So the results from our nutrient analyses actually showed that there was a strong negative relationship between log chlorophyll A and both total nitrogen and total phosphorus. So as both of those two increased, the chlorophyll A decreased. Further, our nutrient analyses indicated that Lake Johnson's trophic status was somewhere between eutrophic to hypereutrophic. However, we couldn't use our total nitrogen to total phosphorus ratio to infer any sort of limitation because the supplies of both were just too high, which makes them non-limiting. So our phytoplankton group presence actually didn't match the published descriptions of phytoplankton seasonality. The diatoms stayed relatively minor throughout our assemblage, and instead the greens were dominant. So this just goes back to what we expected to see: lots of diatoms in the winter, chlorophytes in the spring, cyanobacteria in the summer. Instead, what we actually saw were greens dominating in the winter, and then both chlorophytes and cyanobacteria present in the spring. So in the summer, it's safe to assume that lots of cyanobacteria will be present. We also saw unexpectedly strong negative relationship between chlorophyll A and total nitrogen and total phosphorus. And this could be because of that large rainfall event in early February, which could have flushed nutrients from the reservoir before the phytoplankton could get to them to use them. And the rain also could have just simply diluted the phytoplankton assemblage. And finally, we determined that Lake Johnson's trophic status was eutrophic based off total phosphorus levels and hypereutrophic based off nitrogen levels. And the supplies of both of those nutrients were just too high to be limiting for any naturally occurring phytoplankton assemblage. And then also just going back to our sampling weather, it should be noted that this winter was unexpectedly warm, and our average temperatures were often too high to be optimal for diatoms. So with climate change bringing about warmer weather, it's probably going to become more common to see greens and cyanobacteria earlier in the year. So then quickly, just to hit on some pitfalls, I think that our limited sampling time actually limited us from getting the full picture of this reservoir's true assemblage. And then also there was some human error or my error that could have occurred that could have skewed our results. And then again, I mentioned that large rainfall that probably skewed our results. So if I were to repeat this study in the future, I'd like to see a longer study done with more samples for Lake Johnson, and then perhaps even other North Carolina lakes. So acknowledgements, I just want to say a special thank you to the following people: my mentors, Ellie Allen, Joanne Burkholder, all the ladies in the water quality lab, Jenny, Linda, the whole crew, and the Lake Johnson staff. Without all of these people, none of this would have been possible. All right, any questions?
Hi, Nora. This is Dr. Burkholder. Great job. I really enjoyed your presentation.
Hi, thank you.
Thank
you for your help with that?
Thanks. If you could you tell us a little more about how you would like to have gone after the zooplankton component as well as the phytoplankton? I realize the pandemic cut your research off.
Yeah, it definitely did. I just thought it would have been cool if we had more time to look at the shift in zooplankton structure, and if there were like certain groups of zooplankton that we could research and find out if they, fed on a certain algae species more, and maybe that contributed to like why we saw lots of chlorophytes. Like maybe zooplankton don't eat chlorophytes. I just thought it would have been cool to look into that more, but of course we didn't have that time.
Yeah, I think that's a great observation on your part, because there has been some research that's shown that some green algae have such tough gelatinous outer coverings that zooplankton don't like to eat them. So it'd really be interesting to go after that, especially in a highly turbid system like Lake Johnson. I was struck by your map of Lake Johnson. It's certainly not a beautiful blue water system. So there's a lot of turbidity there.
Yeah.
Hey, Nora, this is Aiden.
Hi.
So I'm with Dr. Schnetzer's lab, which is also a plankton ecology lab, and it sounds like you guys are doing some really similar work.
Yeah.
It's really cool to listen to your talk just because I think we have a project going on looking at plankton assemblages in the Arctic. So hearing kind of the cycles that you were looking at versus the cycles that we've been seeing in the Arctic was really interesting. I think you did a really great job.
Thank you. Yeah.
Great job, Nora. Thanks so much. Up next, we have Aaron Durham. He's a junior in environmental science, and his mentor is Aaron Voigt. So Aaron, feel free to share your screen and take it away.
Okay. Hey everyone, my name is Aaron Durham. Today I'll be presenting to you my research on crab habitats and assessing the effective nursery habitats for juvenile blue crabs. I worked with my mentor, Aaron Voigt. So first off, I think it's important that we understand the importance of blue crabs, especially to North Carolina and the fishery industry here in general. Blue crabs are actually the most valuable fishery in North Carolina, and they bring in 8,000 jobs and gross $26 million annually in like fishing revenue. So why is this important? Why should you care? Blue crab populations are actually decreasing and have been for a while now due to overfishing. On this graph, you see the standing stock biomass, that red line. We see in 1995-ish, it has there's like a sharp increase, and then it decreases and almost never recovers over that standing stock biomass after 2005. And that's an issue. The big problem with that is we stopped overfishing. We made a bunch of different practice changes with fishing blue crab populations, and nothing changed. As you can see, we have never risen above that standing stock biomass again. So if we've done our part as much as we can, we've stopped overfishing, what's the issue? The problem might be looking at the nursery habitats for blue crabs. What is a nursery habitat? Nursery habitats are actually a subset of all habitats where juveniles of the species occur. That's just the general definition, often having a greater level of productivity per unit area than others. And specifically, since we're looking at blue crabs, we'll go into their two main types of nursery habitats, known by their definitional name as like classical and effective. For the classical nursery of a blue crab habitat, we'll be looking at seagrass. For the definition of the classical nursery, it's a habitat that contributes the highest abundance of juveniles per unit area to the adult population. For seagrass, it's always submerged underwater, and it's used as a landing zone around inlets like Hatteras and Ocracoke in North Carolina for the blue crabs to come in after they've been out in the open ocean, and they'll settle down in the seagrass before moving further inland. For the effective nursery, we have what I studied, which is the SDH, or better known as shallow detrital habitat. And this is where blue crabs will end up, usually in their adult years, along the coast in the Marshall areas of North Carolina. And the definition would be the habitat that contributes the greatest total abundance of adult crabs, not necessarily per unit area. In that sense, it's a lot less than seagrass, but overall, possibly more. And so then we get into the actual reason we're here, the reason why this presentation is even a thing. What is SDH? That's the question we're looking for. And the next one is, where can it be found in North Carolina specifically? So what is SDH? In order to determine this, we're looking at the characteristics of what it is, such as eroding, decaying marsh, the sponginess of the soil, and the high water absorption that I've become very accustomed to working in the lab, as well as its high organic matter, otherwise known as roots and shoots. In order to determine these characteristics and come up with a testifiable definition, we're going to use things like wet and dry weight, grain size analysis, and loss on combustion. So at the beginning, before I even started work on this project, Aaron was going out into the Pamlico Sound Peninsula and using a older corresponding blue crab population study that started in 1996 and continued through 2019 in order to determine where SDH presence is or where it might be absent. And so this map here was made based on Aaron's efforts as well as that older study in order to determine where SDH is. And so if you're looking at the map, there's a legend in the corner that shows unsure, like, so that's like a maybe SDH, maybe not, and then presence and absence based on the different colored dots. And so this will become more relevant later on, where we'll apply further information that we've learned to make this graph a little bit, or this map a little bit more precise. And so the process is in order to do that, as I said before, calculating the wet and dry weight, which we did by taking cores from the marsh along the Pamlico Sound, taking them back to CMAS where we weighed them in order to get the wet weight, place them in the pans into an oven, drying them for 72 hours, which we determined to be the most amount of time, or the minimum amount of time needed to completely dry out the soil, and then measuring them again to get their dry weights. Grain size analysis was done with sieves. We just took the soil samples, sieved them through, and separated them into these different baking pans that you see, putting those into the oven and drying them as well in order to get their dry weight. And we have the different measurements in the picture on the side. And for the loss on combustion, pretty simple, just taking a subsample of the dried soil core, homogenizing it, breaking it down with the coffee grinder, and then placing it in a muffle furnace for 4 hours at 500 degrees Celsius. And these are the graphs for the wet and dry weights that we received after calculating all of the information. An average of 69.19%, the standard error 1.74, and then min and max values as shown, as well as the confidence interval of 3.415. And for the grain size analysis, the blocks outlined in red are where we want to draw your attention towards. The largest groups are the roots and shoots, which are in that 4.5 millimeter, as well as the peat-like soil in the 400 micrometer, and then the silty material that's in the 63. And the loss on combustion results with the min of 2.56 and the max of 14.50. And then we want to draw attention towards these outliers that you see and the importance of that. The high outliers are most likely due to some samples having higher levels of organic matter. As we talked about before, SDH is known for its high roots and shoots or organic matter. And these organic pieces of like roots would cause less compact samples, which would vary the weight even though the volume was the same. So that's most likely why we got those high outliers. And so in conclusion, SDH has a 69.19% water weight loss, lots of organic matter, and then the 2.56 to 6.56 majority lost on combustion. And this information here is pretty much what we'll use, or what we use as, I guess you could say, a rubric for determining what is SDH and what's not when going through and processing our 2019 cores. And so now we get back to the maps that I was talking about. So where can SDH be found? We took cores at 38 sites along the Albemarle Pamlico Peninsula and then using those, taking them back to the lab and doing the 3 tests that we talked about, we were able to determine where SDH is or isn't or might be in order to make this older map a little bit more useful. And so this right here is like the culmination of all this beautiful information. These 2 maps are what we're using as our guideline. And so why are there 2 maps? So let me get into it. So for the min and max on the left, you could say this is a more liberal interpretation of where we believe SDH to be or not, with less restrictions on what is and what's not. And so that's why you see so many of these pink dots that determine SDH presence. On the right, there's a 95% confidence interval, which would be more conservative, more strict. And these are based on our data that we use to determine the dry density and the percent water calculations of SDH. And so that's why we get these different variations, because the guidelines for the 95% confidence interval versus the min and max are different. So they give us different variables, which when put into this map form, give different destinations of where SDH might be or not. But in the end, it comes down to preference, which one you want to use. Both are accurate.
Hey, Aaron, I have a question for you.
Hey.
Could you go back to the slide with the 2 SDH maps? Yeah, exactly. Oh, the other one. Yep. So you were saying that the one on the left was calculated based using just like the min and max, and it tends to be like more liberals because you see more areas that are classified as being SDH. Versus the 95% confidence interval one on the right, you see a lot less areas, a lot more in that maybe SDH area. I actually have 2 follow-up questions. What did you use to, what's the maybe? So how did you determine if it was, if it wasn't, or if it was a maybe?
So yes or no is determined based on the dry density and percent water calculations that we calculated. Sorry, I'm losing on my words. But so we calculated dry density and the percent water calculations, and then based on where they fell in along with the 2019 that we used as our guideline, determined whether or not it could be considered SDH based on like that rubric, you could say.
Okay. And then you were saying that you could kind of use either one of these maps kind of based upon what you were looking at. Could you kind of give an example of like why you would want to use one map opposed to the other, like what the, I guess, like benefits and drawbacks are to using each definition?
So for me personally, I would probably go with the 95% confidence interval map, just due to its, I guess, like, or it's more stringent on what is and what's not. And so in that case, you would be less likely to end up at a site and then realize that it's actually not SDH, less so than with the min and max, where a majority of them are considered SDH. So you could go to one site and then it's not really what you were expecting.
Okay, awesome, thank you.
So I've got kind of a follow-up on these 2 different maps. I'm thinking from kind of a conservation perspective, right? If we're about, you know, if these are the important crab habitats, if we were interested in protecting these areas to make sure, you know, that our fisheries are sustained over time, which map might you use to actually decide policy-wise on what areas you would want to protect?
So I'm not, like, I haven't really looked too deep into this, but I would say that a lot of it would be just based on human nature. A lot of it would be based on like typical fishing spots, and then possibly what areas are near boat docks that have like high traffic, because some of these areas are a lot more remote, and we would go to these sites and there'd be nobody there, or there'd just be no reason for anybody to be in that area, and we might be the only boat in that area. And then some of the SDH sites were a little bit more heavily trafficked, or you might see people fishing in that area. And so I think a lot of it would come down to just commercial wise, like what's like, is there another type of like marine species or aquatic species that's being fished in that area that it would just be better to just leave it how it is? Or is this an area that's not really highly trafficked by any type of commercial fishermen, so we can go ahead and block it off and say, yeah, well, this is a protected area. You're not using it anyway, so it doesn't matter. So I think it would be based more off of what would be profitable in that sense, more so than anything else.
Cool. I was thinking maybe the map on the left would be useful if it's like the, you're using a broader definition, you know, or a broader range of sites. So if you wanted to protect the maximum amount of habitat, you might use kind of the broader range. But that makes total sense what you said of incorporating also which of those are actually in use by humans and potentially affected. Cool.
Yeah. And
well, great.
I would, oh, okay.
Yeah.
Oh, I was just going to say, just with the 95% confidence interval, since it's like less likely that some of them are not actually like the perfect SDH habitat, it might be better to use those ones because there's less of them. And so they're more likely to be exactly within that range, just because of how specific the 95% confidence interval, how narrow then it is compared to the minute max.
Gotcha. Yeah. So in a world with limited resources, really focusing, you know, is there. Yeah. Fabulous. Well, thanks so much. If you could stop sharing your screen, then I will turn it over to Phoenix Spivey, our next presenter, who is a senior, also in environmental science, and who is mentored by Andy Maurer. So Phoenix, if you'd like to take it away.
Okay, greetings everyone. My name is Phoenix Spivey, and my project scope covers stable isotope analysis and satellite tracking as a tool for hawksbill sea turtle conservation. So as you can see, there are a bunch of cute little turtles on the screen. These turtles are hawksbill sea turtles. They're named for their hawk-like beak, and their shells often display colorful scutes. And you can basically say they're the cutest charismatic creature. But sadly, the reality for this species of turtle is that they're facing many perils right now. And a lot of these perils are human caused. So some of these distresses include pollution, coastal development, fisheries bycatch, and a decline in quality nesting and foraging habitats. All this together has sort of caused this species of turtle to become critically endangered. So how do we conserve these sea turtles better? Most conservation efforts happen on nesting beaches, but turtles spend majority of their lives at sea. So we need to know where they're migrating after they're nesting to better protect them. And we do this by tracking their migrations. But tracking migrations in itself is expensive. Satellite tools used to track turtles are often like $1,500 a pop. And because of how expensive it is, it also leads this effort to be inefficient. So it's inefficient because of how expensive it is, you get to sample less turtles or track less turtles. But most of all, tracking migrations is important. Once again, it's easier to protect these sea turtles on beaches than it is in the ocean. And once again, we mentioned that turtles spend majority of their lives in the sea. Adult hawksbills spend their lifetime from one foraging home range to one nesting site that they migrate to. And surprisingly, there's little understanding about these foraging sites, key places that we need to know more about so that we can protect these turtles there. So to better conserve them, stable isotopes plus satellite tracking may be the answer. So you may be asking, what are stable isotopes and what makes them a good tracer? Well, the answer lies in you are what you eat. Everything we eat is composed of atoms. As the body grows, it incorporates these atoms into our tissues. These isotopes are stable because they don't decay and they're not radioactive. And isotopes can be heavy or light. Heavy and light isotopes have the same number of protons but different number of neutrons. You can take carbon-13 and carbon-12, for example. Both of them have 6 protons, but carbon-13 has 7 neutrons, one more than carbon-12. Thus, carbon-13 is heavy, while carbon-12 is light. Together, they can form a ratio that is unique in different carbon sources in nature. And as a result, We can use isotope ratios to trace where in space and in the food web animals are getting their energy. So, stable isotopes, along with satellite tracking combined, is overall a less expensive method. It allows for more sample workbook kits when you conduct your study. So you basically get to, because it's cheaper, you get to sample more turtles. Specifically, satellite transmitters can track turtle migrations to foraging and nesting habitats. Skin samples from turtles contain stable isotope ratios that come from what they are eating and from their foraging habitat. So, combining these, researchers have assigned isotope ratios in turtle skins to the satellite tracked foraging locations. Sampling and completing this process multiple times will enable us to identify more points along this space, which leads to the creation of a map of ratios, which can otherwise be called an isoscape. This map will allow conservationists to estimate turtle foraging areas without the burden of expensive tools to track them. So, our question for this project is, are stable isotope ratios an effective tool to track migrations and inform hawksbill conservation planning? Does isotopic variation correlate with variation in foraging geography as determined via satellite tracking? And so our work took place in Antigua. Antigua is located in the Eastern Caribbean Sea, and it's a part of the Leeward Island in the West Indies. This island contains many coral sites and nesting beaches for the hawksbill sea turtles. And there, one of the organizations that we partner with, the Jumbie Bay Project, the Jumbie Bay Hawksbill Sea Project, has been monitoring the hawksbill nesting beaches in Antigua from June to November since 1987. So for our field methods, we collected skin samples from the hawksbills from 2014 to now. These samples were preserved in a salt solution. And along with that, 22 satellite transmitters were also deployed over the past 4 years to track their post-nesting migrations. In the lab methods, this is where I come in. Yeah, definitely spent up to plus 70 hours taking these samples and preparing them for isotopic analysis. We dry the samples in an oven, and like a chef, we chop them into very fine, small, homogeneous particles. We definitely make sure to disinfect before going on to the next sample. And then after the particles were weighed into a tin cap, about 1 milligram, and this tiny cap was turned into small little balls, as you can see over here, and placed in our sampling tray, where we sent it to Yale's Stable Isotopic Lab to do an analysis of carbon and nitrogen isotopic ratios. And so unfortunately, with this whole pandemic going on, they weren't able to do the analysis for that. So instead of getting results, we're just projecting and predicting what what our results would help us create. So yeah, but with those samples, they would eject into an analyzer and convert the gases through combustion. The gases would have been separated and measured by a mass spectrometer to determine the carbon and nitrogen ratios. And so with these ratios information, we hope to create this isoscape as mentioned before. And once again, isoscapes are maps that isotopic ratios of turtles to their foraging location. These foraging locations were once again tracked by a satellite. And from this map, we can predict the foraging habitats of hawksbills throughout the ocean without having to satellite track them. So on this slide, you can see this is an actual isoscape from a loggerhead study that was conducted on the Northwest Atlantic. So that's what that looks like. If you look at the tiny black dots, those are actually the satellite tracked migration points. And so everything in color is just those ratios identified and overlapped over those points. So we hope to get something similar like that. On this slide, we have our migration routes. So this is our satellite tracked data. So you can see that it's satellite tracked information of our turtles from their nesting site to their foraging sites. And so everything in red is their like migration path, and the blue is like their nesting and to the foraging. And yeah, overall, we hope to create a map similar to this. And with this map, we'll be able to allow conservationists to get a clearer idea of where these sea turtles are. And overall, this would allow us to better protect the sea warriors that we love. So with that, I'd like to give a special thanks to the Jumbie Bay Hawksbill Project, Andy Maurer, my mentor. Thanks, Dr. McKinney, Yale Lab for the analysis that they will conduct in the future. And thanks, Bobby, for helping me chop all of those samples. It was a lot of samples, so thanks, everyone. Any questions? Okay. Oh, should I stop sharing screen? Or?
Yeah, that'd be great. That way we can see you. Then
Okay.
This is the time when I invite and remind everybody that you can use the chat feature to type up a question if you want, or just unmute yourself and call out a question one at a time. Thanks, Phoenix.
Hey, Phoenix, I have a question. This is Erica.
Hi.
No, excellent job. Great presentation. But I was curious if there are, you showed a map of loggerhead sea turtle isoscapes. Are there differences in the, like, between loggerheads and hawksbills that make it like easier or harder to do this for the species, the different species?
Yeah, that's an excellent question. So loggerheads have a different diet than hawksbill sea turtles. They tend to eat a lot of seaweed, which has like less variation when it comes to their ratios. And our hawksbills tend to diet, like they tend to eat more sponges, and those sponges in itself has more variation. So like their method definitely works, probably due to their loggerheads diet. Ours, we're trying to see if that will actually work. And we are trying to see if there's going to be an actual pattern, since the variation might kind of interfere with that. So it may not work out the same, but we're hoping so, that the variation won't get in the way.
Yeah, cool, thanks.
Thank you.
Yeah, I've got a quick question for you, Phoenix. Do you, because I don't know much about hawksbill sea turtles except for, you know, what you shared. So do you have any reason like to predict specific patterns? You know, like do you know whether turtles in specific habitat areas or in specific locations are actually foraging for different things? Either because of availability of those different food items, or, you know, is there a density dependent difference in what those animals might be eating?
Gotcha. Actually, I'm not sure if there's a pattern in the stuff that they're eating. I just know that they go for sponges majority of the time. I did know, or I think I did see somewhere that they can resort to other diets or other things to eat, and maybe that could be in relation to like availability with sponges and stuff, but specifically I don't know, other than the sponges that they're already eating.
Cool. I didn't know if the sponges might be kind of an environmental marker, right? Since they're filtering things and picking them up. So maybe they'd get a different signature depending on those different geographic regions. So it'll be really exciting to see what your data, you know, how the data comes out.
Yeah, I am excited too. Thank you.
Well, thanks so much, Phoenix. Up next, we've got Timber Feltz, a senior in international studies, who's mentored by Karen Cooper. So Timber, feel free to share your screen and take it away.
Okay, hi everybody, how's it going? I'm gonna try to figure out how to get this set up. Okay, can everybody see my screen?
No, not yet.
Okay.
There you
Okay.
go.
Awesome. Okay. Hello again. My name is Timber Feltz, and as Aaron mentioned, I'm a senior in international studies. So I kind of wanted to talk about that first. I worked with Dr. Karen Cooper, and we kind of wanted to show the intersectionality between my major, international studies, and how it can relate to applied ecology. And that's where citizen science comes into play. We really just wanted to express the interdisciplinary nature of ecology and expressing how it can relate to some more global goals, specifically the Sustainable Development Goals. So that's where the United Nations 17 Sustainable Development Goals comes into play. And since these goals themselves are so broad, I kind of just wanted to focus on one specifically. So I chose Sustainable Development Goal number 6, clean water and sanitation. And this goal is really fitting because I'd already been working with Dr. Cooper and Amani Bell with the Crowd the Tap research initiative. I'll talk more about it later, but just to give some background, Crowd the Tap is a citizen science initiative with the goal to create a national inventory of water pipes in the United States in order to determine where lead pipes are still located. Basically, I wanted to see how citizen science projects like Crowd the Tap could help us reach United Nations Sustainable Development Goals. And I chose to use Crowd the Tap as a case study for my research, which also kind of helped me bring these global goals down to a local level to make it more relatable. And so just to get more into my research specifically, my hypothesis is citizen science projects such as Crowd the Tap ensure data monitoring of Sustainable Development Goals, and specifically Sustainable Development Goal 6, can be done more efficiently and with less resources than through traditional methods of data monitoring. I'll talk a little bit more about traditional methods and data monitoring later. But right now I want to talk about my predictions. So for the first one, I said that citizen science promotes community engagement, which is necessary in order to meet Sustainable Development Goals. Basically, meeting the Sustainable Development Goals requires an all hands on deck situation. Such ambitious goals cannot be met without the help of as many people as possible. For my second one, I said community engagement through citizen science addresses the gap between setting global goals and achieving them locally. So this is especially true with Crowd the Tap because property owners are responsible for the private portion of their service lines, of their water service lines, and their home plumbing materials. And because of this, public engagement is necessary in order to meet Sustainable Development Goal number 6. Otherwise, many property owners are unaware of where to locate their service lines, and so we can't really receive the data from that if people don't know where to go or if they don't have anywhere to enter this data. My last prediction was through the assistance of non-traditional sources of data monitoring, meaning Sustainable Development Goals will be less costly and more research efficient. Most citizen science initiatives are made up of volunteers doing this work for free, whereas many government or corporate agencies that report data require a large budget to monitor data. Now I want to talk more about my research specifically, and since I'm not really from a science background, it's kind of non-traditional, I guess, in the sense of applied ecology. So I decided to do a literature review, and I wanted to talk specifically about 2 different pieces of literature that I was looking at because they fit pretty perfectly into what I'm trying to say with my hypothesis. The first one was from an article by Veronica Herrera, and she says that over-aggregated indices of water and sanitation access under the Sustainable Development Goals failed to capture much of the variation in service quality, quantity, ease of access, and sustainability of water and sanitation management in developing countries. So she's basically talking about the difference between achieving global goals and how we can apply that to local realities. Then the second one was from an article by Fritz and others. And they say that success in meeting the Sustainable Development Goals will require leadership from the United Nations, innovation from national statistical offices, and focus from the citizen science community to identify the indicators for which citizen science can make a real contribution. And this article is basically saying that we can ultimately improve the process of reporting Sustainable Development Goals through citizen science. Now I want to talk about my case study, which is Crab Tap. It's an EPA-funded project that promotes access to safe drinking water by assisting individuals and groups in creating inventories of pipe materials that deliver drinking water to homes. So this was the perfect case study to use for my research, since it was already kind of focused on a sustainable development goal without really meaning to be. So it's kind of taking this idea of local realities and community engagement support and using it to create a simple way for homeowners and renters to find out what their water service lines are made out of. And I'll show a picture of the kit that we use, or that Crab Tap uses, to help people learn where these materials are and how to figure out what they're made out of. But now I want to talk more about my findings. This graph is showing the gaps between Sustainable Development Goal data coverage and data openness. And data coverage refers to data availability in the last 10 years for each continent that you can see on the graph. And data openness refers to data accessibility. For example, is the data able to be read in a machine readable format, and so on. So it makes it easier to be accessed. I think it's interesting to look at the gap specifically between data coverage and openness in North America and Europe, which according to this gap is the ideal data coverage and openness that we want to reach. And that's one thing that Sustainable Development Goal research doesn't really talk about a lot, is meeting the SDGs in relation to developed nations, because they talk a lot about developing nations. And so I just thought it was interesting to see how much further below we are than Europe. And then this slide, I wanted to talk about how the individual targets and indices that you can see below the general Sustainable Development Goal are used in order to provide a way for countries to use a step-by-step process with a measurement in order to meet the Sustainable Development Goals. However, these are still mostly implemented by the national statistical offices, which are the traditional ways of reporting that I was talking about earlier when reporting the Sustainable Development Goal data. It's important to use these and other non-traditional methods of reporting, especially regards to citizen science data. The literature also reemphasized the importance of engaging local communities and governments, because these global goals cannot be met without addressing the differences between local governments across the globe. And then this slide, I wanted to speak more specifically to local findings. I mentioned earlier that Sustainable Development Goal research doesn't really speak much to what's happening in developed countries. However, thanks to Dr. Cooper's Environmental Science 400 class in the North Carolina Department of Environmental Quality, research has been done on the lead present in North Carolina water systems, demonstrating that government-owned service lines have higher amounts of lead present. Privately owned utilities are more costly, thus people usually people with lower incomes are typically the ones to have their service lines connected to the government-owned utilities, meaning that they're more likely to deal with the effects of lead poisoning. This graph also only shows the water systems that government and private entities have access to, and doesn't necessarily take into consideration water service lines that are located in people's homes, which is why projects like Crowd the Tap and citizen science provided data is necessary. The global implementation of Sustainable Development Goals requires the assistance of several different government entities in collaboration with the member states of the United Nations. The main objective of the Sustainable Development Goals is to provide a common goal that allows all member states to contribute to the global peace and equality that the United Nations represents. That means that the most important part of the Sustainable Development Goals or achieving them is data monitoring. We can't determine if we are meeting the targets for the SDGs without this data. We also can't collect this data without the help of local governments and communities. Different local governments have different ways of monitoring the Sustainable Development Goal targets, and there are differences in the knowledge that government and private utility companies have regarding their water infrastructure. So this makes it difficult to identify a common form of data monitoring. And this is especially true when property owners are involved, because government and private utilities usually don't know what's located on a homeowner's property. For reasons like this, it's important to engage the community instead of relying heavily on national statistical offices, since they don't always have all the correct information. And this is where projects like Crowd the Tap come into play, because they can bridge that gap between what I've been talking about the whole time, between global goals and local realities. So projects like this are also really good at teaching homeowners and renters how to locate their plumbing and water service lines in order to determine the materials they're made out of. Because government entities such as North Carolina DEQ don't really know this. So you can see the kit that is handed out, and it shows people how to determine what their pipes are made out of. And also, if you go to the website, it can help people figure out where to find these pipes. I also wanted to talk about SciStarter because it's such an important tool for citizen science, and also because that's where Crowd the Tap data is entered. SciStarter is an online community that connects people to citizen science projects, and data entered for Crowd the Tap is entered through SciStarter, and people can also easily find projects like Crowd the Tap by searching for projects that they're interested in or just projects that are in the area. While SciStarter is a way to present the endless possibilities of citizen science, each individual citizen science project is different. But local engagement addresses the issue of global goals with local realities, which is why citizen science is necessary. I also wanted to talk about the challenges in doing my research. The first one is that the broad scope of the Sustainable Development Goals makes it difficult to assess all 17 goals, which is why I focus specifically on number 6. But I think that this can be addressed globally by allowing people to focus on research and skills that they've already been invested in. For example, NC State already has a project addressing lead in drinking water, so it makes sense for this project to work specifically on addressing Sustainable Development Goal number 6. And the second one is Sustainable Development Goal research focuses on developing countries, which makes sense, but I think that it's important to look at what developed countries are doing as well. And then we also have the last one, which is research on water and sanitation. There's a lot of research on citizen science and the Sustainable Development Goals, and a lot of research on water and sanitation. However, there's not a lot of research that combines the two together. And I was able to remedy this by connecting it to research being done by Crowd the Tap. However, it seems that research on using citizen science to monitor Sustainable Development Goals is more broad, which makes it more difficult for researchers and citizens to understand the specifics of applying citizen science to the Sustainable Development Goals. I think that future research could address all of these goals by using citizen science. And now I want to say thank you to Dr. Karen Cooper, who is my mentor and a citizen science advocate, and Amani Bell, the graduate research assistant who is working on the crab tap, and also to Dr. Cooper's Environmental Science 400 class and North Carolina Department of Environmental Quality for using their data. Now, if anybody has any questions, I would love to answer them.
Thanks, Timber. Great job. If you could stop sharing your screen, then we'll be able to see you.
Awesome.
And feel more interactive if anybody has any questions.
Of course.
Well, if there aren't any questions, then we'll say thanks again, and we'll move to our last talk for the day, Tessa Pierbolt, a senior in Fisheries, Wildlife, and Conservation Ecology, who was mentored by Jesse Fisher. So Tessa, feel free to share your screen, unmute yourself, and take it away.
All right, give me just one moment while I figure this out. Can everyone see it?
Yep, perfect.
Okay, awesome. I just want to take a moment to thank everybody who is tuned in, as well as congratulate everybody who presented today. You guys did an awesome job, and I'm so excited to see where you guys go in the future. As Erin said, my name is Tessa Pierbolt. I'm a senior in Fisheries, Wildlife, and Conservation Biology, and I'm going to be talking about the characteristics of the American eel in Puerto Rico. Not much is known about this topic, therefore it's kind of a new frontier. To my knowledge, there's only one other paper that mentions the American eel in Puerto Rico, and some of our data is actually featured in this paper. The picture to the right is me holding an American eel in Morehead City. When I joined the project, the data had already been collected, so this was the first time that I actually got to hold a live American eel. We will be looking at life history characteristics such as size, structure, condition, sex ratio, age structure, growth, and mortality. This is a map of Puerto Rico. Most of our samples were taken from two different rivers, the Arcibio River and the Mimays River. I'm sorry if I butchered that. I do not speak Spanish. The American eel is a contagious species. This means that when the mature American eels migrate from freshwater coastal rivers, they go to saltwater and specifically the Sargasso Sea in order to spawn and then expire. They have an extensive range from Greenland all the way to Brazil, and pictured here is the migration patterns from the Sargasso Sea to North America and South America. For a long time, it was thought that these stages were different species of eels instead of different parts of a single life cycle. Each female will release approximately 30 million transparent eggs that will hopefully grow into a fully mature American eel. Once the eggs hatch, the American eel's life cycle starts with the leptocephali. During this stage, they are large, transparent, and flat. They then transition to the glass eel. In this stage, they're still transparent, but are condensed down to look like a tiny eel. As a glass eel, they will begin their journey towards freshwater. Next comes the elver stage, where they gain their coloration. After the elver stage comes the yellow eel stage. In this stage, they basically triple in size. Lastly, we have the silver eel stage, where they are fully mature and ready to swim back to the Sargasso Sea to spawn once and then expire. Our objective is to describe the life history characteristics of the American eel in Puerto Rico. We want to know, are the characteristics of the American eel in Puerto Rico different than the characteristics of the American eel found in North America? Our predictions are that they will be larger at younger ages, they will have shorter life cycle due to being larger, younger, and they will have transition and metamorphosis checks in their otoliths. Pictured here is the life stage of the leptocephali. Personally, I think they kind of look like a scary creature from the deep. For those who are not familiar, an otolith are a very small bone located in the ear of the American eel, as well as many other creatures, including humans. Otoliths are the trio of bones in your ear that vibrate to help you turn the vibrations to the sound of my voice while I'm speaking to you. Otoliths put down rings called annuli, which are similar to the rings of a growing tree. We can count those rings to get the age of the eel, as well as identify transition and times of metamorphosis. Picture here, we have on the left-hand side, we have an American eel otolith in a sugar cube sized mold of epoxy. At the top, we have the reading process of American eel, where we can see the annuli and the defining features. On the right is a size comparison with a penny. On the right-hand side of the penny is a largemouth bass, and on the left side of the penny is a bluefish. I would like to note that American eel otoliths are much, much smaller than the bluefish. We collected paired American eel otoliths from Puerto Rico between June and July of 2015. Otoliths were mounted onto slides via thermoplastic cement and polished, as well as mounted in epoxy and cross-sectioned. Otoliths were read by 2 readers to determine a consensus age. We strive to calculate size, structure, condition, sex ratio, age structure, growth, and mortality. Some of these calculations are unfortunately pending due to COVID-19. Here we have a picture of the microscope we used, the picture of the otolith and epoxy, our wonderful map of Puerto Rico, as well as an example of how we put the slides and the otoliths and the thermoplastic cement all together so that we could read the otoliths. So we had a sample size of 120 eels with a subsample of 94 eels, which consisted of the eels that were sampled for otoliths. We did see that many of the eels had transition and metamorphosis checks on their otoliths. Out of our sample of 120 eels, we had 9 males, 34 females, and 77 unidentified. Even though this is a small sample, we did have a lot of unidentified eels. This still shows that their sex ratio is very skewed, and this causes major issues for the population down the line. The first graph is the graph showing the mean length at age, and our second graph shows how many sampled eels were at each age in years. As we can see, most of the sampled eels were around the age of 3. From these results, we can see that the eels are larger at younger ages. We can conclude that they are maturing at earlier ages and therefore are transitioning through their life cycle at a quicker pace. There is very little published on the American eel in Puerto Rico, so this research can be used as a baseline to further American eel studies in Puerto Rico. Some of the challenges that we faced was, since there was little on the American eel in Puerto Rico, we were kind of flying the ship blind, and we had lab time cut short due to COVID-19. We have a picture here of an American eel predator, the river otter, having a little snack. I would like to thank my research advisor, Jesse Fisher, for all his help and support. I would also like to thank Spencer, Dr. Kwok, Mr. Laney, Cliff, and Carl for all the work that they put on this project as well. And I would also like to thank Erin for all she has done to help me out, and as well as putting together this event. Here are some citations. And I would like to comment that these are actually conger eels and not American eels, but I'm ready for questions when you guys are.
Awesome, thanks. Great job, Tessa. And final call for Q&A, opening it up on the chat.
Hey Tessa, Andy Maurer here. Great talk. Thanks for sharing all that work that you did. I was wondering if you could elaborate maybe just a little bit more on the results you showed that eels are growing longer earlier, I believe, and I was wondering if maybe you had some implications in mind or what that means for the eels and their ecology.
Right, thank you for your question. So basically what this means is that we think because we're at a warmer climate, that the eels are getting more nutrients or something is being put into that to make the eels grow quicker at a younger age. Most fish species have indeterminate growth, so it all depends on how much nutrients and their environment to determine how big or small that they're going to get. So basically this means for the population is that they're going through their life cycle quicker, which means that each stage usually takes a certain amount of time. So going from the eggs to the leptocephali takes so long, going from that stage to the next stage. So basically because of something in their environment, they're speeding through this cycle, which is causing them to mature and spawn at earlier ages than those of the those that are found here in North America. Another thing that's super interesting about this is that I also looked at Carl Hintz's otoliths that he has for the Roanoke American eels in Roanoke River. And when you look at the otolith sizes along with the different structures within there, you can completely see a difference. The otoliths in the American eels are much smaller than the Roanoke eels. And so we would need to do more analysis and data collection to kind of figure out what are these elements that are causing these different things to happen at these places. Did that answer your question?
Yeah, that was great. Thanks for the answer and great talk. Thanks.
Thank you for the question.
Tessa, I'll let you off the hook so you don't have to sit waiting. But thanks again to everybody who came, who participated. Thanks to all of you students for presenting your research and for the hard work you've done over the past semester or year, especially in the face of these new exciting challenges. And thanks to everybody who joined us from the comfort of your own home. I think this was a pretty exceptionally successful online transition to our minor research symposium. So with that, I'll echo what folks are saying in the chat. Congratulations to all of you, really excellent talks. You've taught us so much about a diversity of fields of study today and study systems. So y'all take care, stay healthy, and we'll see you around.